So thank you all for coming to watch my presentation. I'm excited to talk to you about some experimental evolution work I've been doing with rhizobia genomes. So bacteria genomes vary across species. I'm particularly interested in rhizobia. These are bacteria that can live on a plant host, so they exchange nitrogen for carbon inside root nodules. That's pictured there on the right plant picture. And a common feature of rhizobia genomes is that their genomes are often split into different replicons, and one hypothesis is that functionally different genes will be clustered on different replicons. So maybe all of the genes involved in symbiosis will be on one replicon, where genes will be on the other replicons. So if we look at this rhizobia, this blue rhizobia as an example, these are some components you might find in their genome. So the chromosome is usually the largest replicon. It contains genes that are essential for growth. I'm going to skip over to the megaplasmid and plasmid categories. These are smaller replicons. They might not have essential genes for growth, and they could be laterally transferred into the cell. And then a chromid can kind of have characteristics of both chromosomes and plasmids or megaplasmids. And then megaplasmids are larger than plasmids, and that's the main difference there. So I'm particularly interested in this one species of rhizobium, Rhizobium meliloti. This is what its genome looks like. So it has a large chromosome, 3.7 megabases. This has all the core genes or housekeeping genes necessary for growth. And then it has a PSIM-B chromid. This is an evolutionarily older chromid. And another key feature of it is that it has some diverse metabolism genes on it. And then finally, the smallest component is this PSIM-A. Hopefully you can see at the yellow writing. This is an evolutionarily younger megaplasmid compared to PSIM-B. So let's go through these components. So over time, there's been gene transfer of some core genes from the chromosome to PSIM-B. So there are this chunk, or we have evidence of one chunk in particular going on to PSIM-B. We expect that there could still be some gene transfer between the chromosome and PSIM-A, but we don't have concrete examples of that. So PSIM-B is older. There's been this transfer of core genes. Both PSIM-A and PSIM-B have symbiotic genes that are necessary for life on a plant host. So that's depicted in green. So on PSIM-A, you have the nodulation and nitrogen fixation genes necessary for development of nodules. And then you have exopolysaccharide genes as one example of some symbiotic genes on PSIM-B. So these genes are also should be important for nodulation development. Okay, so this is the overall genome. Oops, sorry, shaking a little, the podium's shaking. Okay, so these are the components of the genome, but what's another thing that's interesting about Rhizobium meliloti is that it can live in multiple different habitats. So it can live on a plant host in a nodule, it can live in the soil, we can also culture it in the lab in media. So how do these different components matter in these different environments? So when you're living in a nodule, the PSIM-A and PSIM-B replicons will be important. We need those genes to establish this relationship. And how we think that these genes developed on these other replicons is that when a rhizobium is first introduced to a plant host as a new environment, there's selection for genetic change to happen on these other replicons other than the chromosome, because any rearrangements to the chromosome and core genes could be costly. So that's how we end up with these specialized replicons, PSIM-A and PSIM-B, that are really good for life on a plant host. So then when we move the rhizobium into a different environment, so we're moving it into the soil, here we still need PSIM-B because it has these core genes that it got from the chromosome, so it will be important in this environment. But PSIM-A, which is specialized for a plant host, could be costly in this environment. So it's costly to maintain this extra genetic material or this replicon when there's no plant host around. Same when we think about this rhizobium living in media. There's no plant host around, so it could be costly to have PSIM-B around in this location. So previous work has developed these 4 different strains, or constructed these 4 different strains where we have the presence and absence of PSEM-A and PSEM-B so that these kinds of theories and questions could be tested. So how they were made was PSEM-A and B were first taken out of the cell, and then either both were put back in, so that's the top strain, just PSEM-B was put back in, the second strain, both were left out, or just PSEM-A was put in. So that's how they were constructed. I did mention that there are some core genes on PSEM-B. Those were taken out and put onto the chromosome so that we could get a strain that could grow without PSEM-B. So just a few genes were transferred over. I'm going to use these labels for the rest of the presentation, just for some simplicity. And then I will use this color coding, which hopefully shows up on the slides, and then you can see. Okay, so in previous work, these 4 strains were grown in a TY media to see how having these extra replicons could matter. And you'll notice that the 2 strains without PSEM-B are limited in their growth in TY media. And we know that this is because these strains are limited in the amount of, or the different carbon sources they can use for growth. We think because it's missing that PSEM-B plasmid, which has these diverse metabolism genes. So they don't grow as fast and they don't grow to as high density in TY. So what I wanted to do was, taking these strains, I wanted to do an evolution experiment where we try to adapt these strains to life growing in a TY media. Our recipe is there. We're giving it sucrose and yeast extract, which should have some carbon sources for growth. We want to evolve it in this media to see if we can recover the growth rates in those 2 bottom samples where we don't have PSEM-B. So even though it doesn't have PSEM-B, if we evolve it in this media, can we increase its overall growth rate? And then identify genes with mutations that are associated with that phenotype. So I don't have any genetic data today, but I can show you the phenotype part. So this was our overall experimental setup. So we have the 4 different strains. We had 5 replicates of each. We grew it in test tubes with this TY media. We grew them to stationary phase, and then we transferred 1% of the volume over to a fresh tube. And continued that. So for the strains that have PSEM-B, this meant there was about 8 bacterial generations every time we did a transfer. And for PSEM-A, it was about 7. So we continued, or repeated this process until all 4 categories had been through around 200 generations. Then we plated them out, we picked different colonies for streaking, and we ended up with 25 strains from each category that had been through this evolution process to test. Okay, so here, this graph, I'll show you the results. So on the x-axis, this is the time in hours. On the y-axis is the OD600 reading, so our measure of bacteria density growth. So all of the ancestor strains are in this bottom category. They have the same color code as before. The evolved strains will be a similar color, but just a lighter shade, so you might not be able to see it, so you'll just have to trust me where they are. But in this case, and I'll also have the diagram of the cell showing what the strains were. So these were bacteria cells with both PSEM-A and PSEM-B. So in this case, all the evolved strains that had both of these replicons, they actually performed, or they look similar to the ancestor. So all of the light blue lines, which you can't see, they're actually all kind of around that dark blue line anyways. So you'll just have to believe me. And so then when we compared each evolved strain back to the ancestor, they were not significant in terms of doubling time or in the maximum density that they grew. There was one that didn't grow as much, so it's very light, it's a little bit lower. But I want to retest that one just to make sure that it's not an outlier. So these strains, they look the same as the ancestor. After evolution, when we looked at the strains that had PSEM-B but no PSEM-A, we noticed, so those are the light pink, hopefully you can kind of see them at the side. All of the strains were significantly higher in terms of doubling time, so they increased in terms of growth rate. They did not change in terms of maximum density that the bacteria were reaching. So we do see some improvement in growth rate. Then when we look at, now we'll go into the strains that don't have PSEM-B. So this is one that have PSEM-A but no PSEM-B. So the yellow lines are kind of, I'll try to point them out. They're kind of like here. So they're not, so when we compared these evolved strains back to the ancestor, they didn't increase or change in doubling time, but they reached a higher density, all of them did. So they did increase in terms of bacterial growth. The same story, hopefully these lines are a little bit easier to see. So these are cell or bacteria strains that didn't have PSMA or B. After evolution, they were all significant in terms of the maximum density they reached in the test tubes. Okay, so overall, our main results here are that the evolved strains without PSMB do show increase in total bacterial density that they're able to reach in our test tube experiments. And we think that this could be that they are evolving capabilities to use more carbon sources in the media, since before we know they were limited by which carbon sources they can use. And we think that's because they didn't have PSMB with these metabolism genes. So now they didn't quite reach the level of growth as the strains with PSMB, but they certainly improved. So next steps will be to identify which mutations are associated with this increase in improved growth. And then something else I want to do is take the original ancestral PSMA and PSMB, put them into the evolved isolates to see if those mutations only matter when the original genetic material isn't there, or like putting these plasmids back in if that just recovers it and those extra mutations don't matter. Okay, so for this work, I'd like to thank my lab and supervisor, George DiCenzo at Queen's University. Everyone listed here has been helpful in terms of project ideas. I did want to mention Allie, because she did some previous evolution experiments, so I used a lot of her protocols. Also want to thank NSERC for the postdoctoral fellowship and a travel award from Queen's that allowed me to come here. So thank you. Yes.
I wondered if those are conjugated plasmids and what you think might happen if you let them move
between the strains. Yeah. That would be interesting, and that's sort of what I am like trying to want to do with the next step is to take the original plasmids and put them back in just to see if, like, now that it has this original these original genes that it had, how that will matter. But in terms of like mixing them in the tube and like looking for like transfer, is that what you were asking? Yeah, that would be interesting. And I think in that case, any ones that have the PSMB will grow to much higher densities in terms of jumping into other cells and then boosting them up too. That could be interesting too. Yeah, so this ties into other work that I'm doing that definitely is more applied. So I do think that when we do evolution experiments in the lab, we are kind of adapting them to be good at living in culture. And then if you want to put them like back on a plant, they might have like lost some of that or those like beneficial background that is good for living on a plant. So I think in terms of these strains here, like only the one with both PSMA and PSMB would be able to make nodules on a plant. I predict that if we did try it on a plant, it would be worse just because it went through 200 generations in a test tube. But that is something that I am wanting to do with other rhizobia, like ones that we want to use as inoculum for certain crops. How can we like evolve them in the lab to be better if we don't want to like keep putting them on a plant, because that can take a long time. So yeah, there's definitely related aspects to that. Does that answer your question?
Yeah, absolutely.
Okay, great.
Hi, everybody. I'm going to present a talk called Finding Adaptations by Evolving Them Away. I work on this really cool microbe called Dictyostelium discoideum, which is really cool for lots of reasons that I could belabor, but for right now, just appreciate that it forms these lovely multicellular fruiting bodies, which are depicted here. Now, Dicty's been studied by lots of people for a long time for lots of reasons, but one of the reasons is it has this really cool life cycle where it's facultatively multicellular. It goes back and forth between a unicellular and a multicellular life cycle. And so the way this works is it spends most of its time on the left there, the vegetative stage, where it's behaving like you probably imagine an amoeba should behave. It's a single cell, it's moving around in the dirt, it's hunting for bacteria and reproducing asexually. However, when it starts to starve, when it runs out of food, the cells will aggregate together. They first form a multicellular slug, which can move through the soil and find an appropriate spot to then form those fruiting body structures that were in the previous slide. And then these fruiting bodies consist of a stalk with a bunch of durable spores at the top. The spores can hang out at the top, wait to be dispersed, and eventually hatch out and re-enter the vegetative stage of the life cycle. And so move back and forth between being a single and multicellular organism. Now, one of the things that Dicty is great for studying is cooperation and conflict, because the formation of these fruiting bodies involves altruism. The cells that are in the stalk die, and it's only the cells that are up in the sorus that retain any fitness. And so this, of course, sets up once again that classical evolution question about how can selection support altruism? However, you might be asking yourself, wait a minute, lots and lots of multicellular organisms have lots of somatic tissues. So if you imagine a more conventional multicellular organism like a human, the vast majority of its cells don't have any fitness and are just sort of serving a tiny minority. So it's similar to that for the Dicty. However, what's special about Dicty is it is an aggregative multicellular organism. It does not come from the clonal replication of a single cell so that all of its cells are related. Instead, it aggregates from whatever cells happen to be nearby. And so hopefully that makes it clear to you how Dicty can end up with lots of chimeric multicellular structures where there's more than one lineage inside of the same body. And this should massively increase the risk of cheaters and the sort of exploitation that makes altruism so interesting to study for evolutionary biologists. And so in Dicty, a cheater would be a strain that doesn't, volunteers not to contribute towards stock formation, lets the other guy do that, and disproportionately is represented in the spores that get to survive. And this sort of cheater, these kind of obligate cheaters, show up all the time in the lab. If you just grow Dicty and passage it in a low relatedness environment, you get these cheaters popping up new all the time. However, they've never been isolated in nature. And there are some researchers who have speculated that maybe all the cheating that we think is so important maybe is not adaptive, maybe is not something that's particularly relevant to Dicty in the wild. It might be kind of a lab artifact, the result of other traits, other life history traits that just look like cheating. And it turns out actually studying Dicty in its natural habitat is really, really difficult because it's really, really small. And so it behooves us as evolutionary biologists to try and come up with lots of different cool ways to look into things like this. And so that brings me to the purpose of my study, which, again, finding adaptations by evolving them away. And this was inspired by the observation that organisms tend to use it or lose it. That is to say, when selective pressures are relaxed, traits which are adaptations to those pressures should tend to be atrophied. And there's lots of cool examples of this, but probably the sexiest are these troglomorphic fish, fish that end up in low light caves and pretty much across the board end up evolving away their eyes and their pigmentation. This has happened in lots and lots of different fish all over the world, and so it's super cool. And so imagine, if you will, a fish. It's a complicated organism with lots of different traits, many of which are adaptations to many different selective pressures. And so, it's under pressure to have a growth rate, it's under pressure to produce lots of offspring, it's under pressure to survive whatever hazards are in its environment. And of course, if any mutations occur which disrupt any of those adaptations, they're going to tend to be selected out. And also, different adaptations may trade off negatively with one another, such that the final fish kind of represents a compromise between adaptations to many different selective pressures. Now imagine you take that fish and you put him in a new environment where some of those selective pressures are relaxed, like, for instance, the need to see or the need to protect yourself against UV radiation. You might expect that the fish would lose these traits and perhaps gain in other traits that were previously trading off with them. And so I want to do the same thing with Dictee. And my thinking was Dictee, similarly to the fish, is going to be under all sorts of different selective pressures, and some of them are going to be specific to these different stages in the life cycle that I described. So it'll have some vegetative stage specific and social stage specific selective pressures. And if cheating is adaptive in nature, if social conflict matters to Dictee in the wild, it's going to be only during this social stage where they're actually making these fruiting bodies. And so in the same way, I want to relax certain selective pressures and see if, like the cave fish, Dictee becomes blind and loses the ability to, loses adaptations related to cheating. So the plan is, I take a whole bunch of different wild Dictee strains, I experimentally evolve them under a condition where they never enter the social stage at all. Super easy to do with Dictee, you just keep them well fed. So they never aggregate and they just stay vegetative amoeba forever. And then ultimately I'm going to be looking for, do they become worse cheaters? Do they become, just in the way that cave fish become worse at I Spy, these Dictee should become worse at cheaters, but only if cheating was previously under selection. And so I'm also looking at a couple of other traits to help contextualize this. And so for one, I'm going to look at the growth rate during the vegetative stage. So this is something they should be selected for in nature, but also selected for in the experiment, and so should not go down. In fact, I think it should increase. And then slug mobility, you might recall that prior to making the fruiting body, Dictee forms this slug that can move around. I think it's safe to assume that that's an adaptation, and it's also something that is not going to be relevant in the lab, and so it's something I would expect, again, like the cavefish eyes or the cheating, if cheating was adaptive, to go away. So what happened? So first, the vegetative stage, vegetative growth. Again, this is something that should be selected both in nature and in the experiment. The evolved lines had higher growth, which makes perfect sense. They adapted to the laboratory environment. They grew faster. For the slug migration, these are assays where, here, I'll skip to this slide. I don't know how well you can see it, but we plate these Dictee up at the top and then put a light source down at the bottom of the plate and see how far the slugs can move. And hopefully you can see the evolved lines, as we predicted, do not move as far, which we take to be they're becoming nearsighted using my fish allegory again. And then so finally to the real question, do they become worse at cheating? They do. So in these assays, we're just looking at what proportion of the spores do they make up in a chimera with a control strain. And you can see they significantly decrease. They get worse at it. So they're either getting worse at cheating or worse at resisting cheating, either of which we take to be support for the idea that they had adaptations to make them better cheaters or better at resisting cheating. And then when we took them away by putting them in an environment where that no longer mattered, they lost them. And so I take this as some piece of evidence to help support the idea that Dictyostelium does undergo social conflict, does need to cheat or resist cheating in nature. And I believe, in general, perhaps approaches like this could be adapted to looking at certain other microbes, many of which have some of the same problems of studying Dictee, of where it's really, really hard to actually look at them in their natural context without putting them in this very artificial lab environment. And I kind of motored through that. I thought I wasn't going to have enough time. But here are my acknowledgments, thanks to my committee, my advisors, everyone in my lab, and all of you, and I would love to take any questions you might have. Gee, I shaved 2 minutes off it. I don't know how I did that. Yeah.
So thank you for your talk. One thing I am wondering when you're trying to find evidence of adaptations to cheating, how are you able to distinguish whether those adaptations were specific specifically for cheating versus for just rock formation that are related to that also involve in cheating? So how do you disentangle those two?
With this alone, I don't think you can, because of course the way that I relax the selective pressure is going to relax all of the social cycle, social stage related selective pressures. I did do some investigating of some other traits, looking for. We were interested, are the dictys becoming worse at making spores? Are there other aspects of their development that they're changing on? And we got sort of mixed results on that. So I don't think with this experiment alone you can disentangle that, and I wouldn't try to claim that this is proof that cheating is definitely important for dicty. But there are other lines of argument that also support that. But yeah, I think in general, if you were designing it, trying to use this concept to look at something else, you'd want to, as much as possible, be able to specifically relax the selection pressure that you wanted, which is tricky. Yes, sir?
That was super cool. Could you just revisit the slide where you showed that cheating had decreased data? What was the measure on the y-axis?
So this is what percentage of the, so in these assays, I took whatever strain I was looking at, whether it be ancestral or evolved, I combined it with a fluorescently labeled control strain, and then at a 50-50 ratio. And so if there's no cheating going on, then I put them in at 50-50. When I then collect them and pick up the spores, half of them should come from my strain of interest and half from the control strain. And so this is just looking at that proportion. And so you can see that in general, in both cases, both evolved and the ancestor are higher than 50%. So my control strain turns out to be less socially competitive than the strains I was studying. But that's not particularly relevant because I used the same control strain for everything.
There we go. Not that one? Well, thank you, everybody. I'm super excited to present these new preliminary results of a new line I'm starting. And I think you all have enough time to read the title, so I'm going to just jump in and say that the background of the research I'm going to be presenting is I'm interested in how dynamic microbial communities function. And dynamic communities are remarkably diverse, genetically, physiological, biochemistry, allowing them to flourish in a ton of environments and hosts. And in recent years, there's been much research trying to understand the capabilities and impacts of these microbial communities in hosts and their environments. However, deciphering these microbial communities by phenotypic signatures is challenging. Our ability to predict and manage the function of these communities is still far from realized. So apart from all of the ecological surveys in which people are sequencing, doing metabolomics, metagenomics, there's been also other approaches experimentally in which they're trying to basically address these questions with experiments. Some of them is to just scoop natural communities in the lab and just like look at them and try to stabilize them. Other approach is what I've called artificial communities, so basically isolating different players, putting them together, and then study them. The approach I'm going to be presented is what they call synthetic communities. This kind of, I was like, okay, where's the definition? And there was like some people that was claiming that for synthetic communities, I don't know if you can see my cursor, but to the left, you actually have to have constructed strains and other definitions in which you just can have a stable community. I'm going to take the second approach, so I haven't constructed anything. And I'm going to define a synthetic community as basically a technique in which we involve co-culturing multiple taxa and in well-defined conditions to mimic the structure and function of like the microbiome that we're trying to study. Now, from all of the possible communities that I could be studying, the one I'm really interested in is like the interplay of cyanobacteria. I'm going to say quote unquote now heterotrophs, and I want to tell you why, because I know now that they're not exactly heterotrophs, and the toxicity of the cyanobacteria. So my question is how the host bacteria, which influence the cyanobacteria population dynamics and the toxin production. And for those that are not familiar with cyanobacteria, and specifically bloom-forming cyanobacteria, they're really large, diverse phylum, globally distribution, really important for biochemical cycles, and they're present both in marine and freshwater food webs, and they are oxygen and photosynthetic prokaryotes. Some of them can form what we call harmful algae blooms, that is like this overgrowth of these cyanobacteria that can alter the ecosystem processes. They also basically can disrupt and produce toxins that are bad for us and for the fish. And they actually are used for biotechnology. So if you have eaten spirulina, it's actually a bloom-forming cyanobacteria, and they can produce toxins too, which I still feel weird about eating them. So recent evidence highlight that there is like a role in these blooms of cyanobacteria of like what I call the co-occurring bacteria. That I don't know if you can see it well, but I marked them with like an arrow down there. But if you scoop a bloom, you're gonna see a ton of bacteria apart from the cyanobacteria in the water. And there is an evidence that those bacteria actually alter the community compositions, and they can drive the succession of the different cyanobacteria in the bloom, and then make them persist more or less, the cyanobacteria, so the length of the bloom. Also, these growth effects that have been highlighted of the cyanobacteria affecting the co-occurring bacteria and making them grow in different parents, and obviously the extracellular metabolites. As I mentioned, I'm interested in the toxicity. So the co-occurring bacteria actually have growth effects in the cyanobacteria, changing how much toxin they can produce. So now I'm gonna jump into the methods. So what I did, I went out and about, found some blooms, scooped them out, and obtained what I'm gonna call xenic cyanobacteria cultures. For anybody that has not worked with these bugs, getting axenic, so monospecies cyanobacteria, is really, really hard. Like, even if you have single colonies and you're convinced you have one, then you play them in a rich media without light, and then something pops up. So when I got like my xenic strain, what I did is like exactly check if there was something co-occurring with them. So trying to isolate and identify what I thought it was heterotrophic species. And then I checked for toxicity. All the toxic analysis that I've done, it was ELISA. I haven't sent it to like an HPLC yet. And obviously, I checked the strains that I was isolating. And what I got after several isolations is different xenic strains of Microcystis, Synetococcus, and stuff I still don't know what they are. All of the Microcystis that I have in the lab right now produce microcystin, which is the toxin. None of the Synetococcus were positive for that toxin. So now that I have them, I passed them for a couple years because I wanted them to be stable with their co-occurring bacteria. And my experimental design was simple. I was like tons of replicates of the Microcystis community, tons of replicates of the Synetococcus community, and then mixing them both. And then I follow them for the first 21 days, almost every day, and then transfers of 21 days. And then I'm gonna jump straight into the results. So what you're seeing here in the graph is basically the meal cell per milliliter, so the concentration of the cyanobacteria over those 20 first days. And you have the microcystin community in the red, the mix in green, and the Synetococcus in blue. And suddenly you can see they're growing, which is good. And the mixed one actually is an in-between both of the pure cyanobacteria cultures. So I'm seeing an effect when I'm mixing them together. One thing I want to highlight, and now this is a complicated graph, but basically I'm going to just jump into the meat, which is I can tell them apart from size. So Synetococcus spigoplancton is really, really, really small, whereas Microcystis is actually bigger. So if I use a culture counter multisizer, what I can tell is like this bigger peak around 3 micrometers of diameter has to be the Microcystis, whereas the Synetococcus has to be smaller. So I kind of have a semi-quantitative trait to follow the mixed population and knowing which of the two cyanobacteria are present. And then I was also tracking the toxicity. The Synetococcus was never toxic. And then the mix actually presented similar toxicity than the just the toxic strain. I know there's a ton of variability in that graph. I merged all the data because like the toxin is intracellular, so I was doing dissolved toxicity and then also breaking the cells. So the moment you break the cells, you get a lot of variability. And I was saying they have things that grow with them. I just wanted to present them. We can follow them phenotypically again by colony formation units. And what I was seeing, it's like actually the dynamics of these bacteria within the populations of cyanobacteria were changing over time. This is the microcystic treatment, this is the Synetococcus, and this is the mix. And one of the things that I sent them all for sanguine sequencing. Most of them were uncultured bacteria. Some of them were like Barioborax, actually heterotrophs, a lot of Pseudomonas, a lot of non-hits, but it was Sanger, so I just stopped there and I'll talk more of sequencing further later on. But what we can see is like, okay, those two that are present in the microcystis is actually present in the mix. These two that are present in the Synetococcus, they're actually present in the mixed population too, but also the mixed populations kind of like, oh, sorry. Can I go back? Let me go back. Oh, kind of like down here is like these two. I was like, I was not able to see them before in the plates until I put them together. So now I know they probably, either it's a contamination, that I hope not, so I repeated this multiple times, or the moment I was mixing them together, some strains that were in low densities actually were able to be in a higher density that I can actually detect them. And then that was the first 21 days, and then I looked through the experimental evolution over 4 transfers. And the only thing you have to focus here is this. So this is my peak of microcystis in a mixed population, is disappearing. So it's being overcome by the Synetococcus. I just want to show here that I was also tracking the bacteria, and some of them actually were picking up. For example, this one here in the mixed population that I cannot tell you what it is because it's one of my known hits, but the dynamic of the co-occurrent bacteria were also changing. So looking close at the mix, which I think is the most important treatment of this experiment, as I said, we can track them phenotypically. So those peaks would say, I'm pretty much losing the microcystis in all the populations of it3, which it was number 3, number 4, and number 8. And that data kind of matched the patterns of toxicity that I'm seeing. So this is the microcystin production. So the non-toxic was not producing anything. All the toxic were producing a ton of them, which would be the red. And then in the mix, again, I marked with red, the populations that were still producing toxicity were the ones that had the microcystis present. So I got all of this, and I did shotgun metagenomics. And what I see here, what you're seeing is basically what I call the microcystis ancestral with the yellow, which is going to be the microcystis, the Synetococcus ancestral with the brown, that went too fast, that is going to be the Synetococcus, and then all the populations after 4 transfers. And basically, the take-home message of this slide, and I know there's a lot of colors and a lot of noise, is like the microcystis are completely different from the Synetococcus, and the mixed one looks super similar to the Synetococcus. So these two basically are not significantly different, except for population 8, which is the one in the corner with some yellow, that actually maintain the microcystis, and I'm really interested to know why that population maintained that species. And I was saying at the beginning, I'm going to call them heterotrophs with quotes, because if you go through the list of all of these bacteria, most of them are non-oxygenic for photosynthetic bacteria, nitrogen fixers that they only have described in soil, and a lot of Pseudomonas, and the Barioborax that is a generalist. And I was like, oh, wow, that was not what I was thinking. I was expecting them to be all heterotrophs. I think it's an artifact of me adapting them to the lab conditions for a year. Just looking at the bacteria quickly, I was checking how they were doing with the toxin. So I was doing growth assays, and I'm just going to show 2 of them. I have a ton of growth assays of all of these bacteria that I isolated. But I got a Barioborax from the microcystis treatment, and what you're seeing here is the growth versus time. And here is in the right, you have basically different doses. And here is the sigmoidal response of the maximum dose. So what you're seeing is actually these bacteria are growing better with the toxin. Whereas the same varioborax, but in the mixed population, is kind of doing the opposite. A lower doses of the toxin is doing better, but then actually the toxin is toxic for them at higher doses. And I've been doing all of this with all the strains. Wait, almost done. And basically checking if they were degrading. My first degrading assays are not giving any significance right now, so they don't seem to be degrading the toxins. So they take home. And when I say take home, these are preliminary results. I think I need to look at way more of these interactions between different communities. So taking with a grain of salt, saying more or less this is to kind of suggest future experiments and future research. But I think the phenotypic focus can inform these temporal and functional dynamics of cyanobacteria and their core-current bacteria. This is not moving. There's kind of a support that is one of the theories that like each cyanobacteria has their own phycosphere. We cannot rule with this data if it's taxa or function specific, but I see some trends towards that, support that direction. And actually the co-occurrent bacteria growth is affected by the cyanobacteria. In this case, we're seeing the effect of the toxin. With this, I want to thank my former lab and the funding that is supporting me, and I'm happy to take any questions. Just to say I'm starting my new lab this August, so I'm recruiting. If you're interested in this or anything with water-related microbes, I'm happy to talk.
So thanks for sticking it out for my talk. So my name's Sarah Worthan, and I am a postdoc in the Behringer lab at Vanderbilt University. So in nature, microbes often encounter the limitation of nutrients in their environment. And nutrients often come in the form of feast-famine cycles. So for E. coli and other organisms that have to transition between different environments in their lifestyle, this can be quite stressful. And this nutrient stress is compounded by changes in other factors like changes in salinity, pH, oxygen, and temperature. And so at this point, keeping this in mind, I'm going to talk about some outcomes that we observed after experimentally evolving E. coli to feast-famine cycles. So this experimental evolution was performed in culture tubes using an amino acid-based broth called LB. And so as E. coli age in this broth, it becomes alkaline. And so not only are these bacteria dealing with nutrient stress, they're also dealing with pH stress. And so these bacteria were starved for 100 days and transferred 9 times for a total evolution of 1,900 days. And when we look at the mutations in the populations, we actually see lots of mutations, but some of the earliest and most parallel are mutations in 2 genes, YDCI and Rho, where 13 out of the 16 populations contain mutations in YDCI, and 7 out of the 16 populations contain mutations in Rho. And what's important is that every population that contains a mutation in Rho also contains a mutation in YDCI. And when we look closer at the location of these mutations, in YDCI we see they span the entirety of the gene, suggesting to us that they probably knock out its function. YDCI encodes a transcriptional regulator that's thought to be horizontally transferred from Salmonella species, where it's been shown to be associated with pH homeostasis. Alternatively, we see a different story with Rho, where all of the mutations cluster in one region of the gene. And in fact, 5 out of the 7 mutations we observed in Rho were all the same arginine to histidine mutation. And so this suggested to us that this mutation may alter the function of Rho. So Rho encodes an essential transcriptional terminator in E. coli. And we were really struck by the co-occurrence of these 2 mutations. We decided to replay evolution, starting from a wild-type genotype, and then looking for the mutant genotypes after 100 days. And still, we were unable to isolate any population that contained only the Rho mutation without a YDCI mutation present. And so we asked ourselves, why would these mutations show up together, and what types of phenotypes do they confer? And so first we started by studying the Rho mutation to try to understand how this mutation could have altered its function. And so in order to terminate transcription, Rho binds to RNA and functions as a helicase as it translocates the RNA strand and unwinds the RNA-DNA duplex, disassociates RNA polymerase, and thereby terminates transcription. And so we looked at one aspect of Rho's activity, its ability to unwind an RNA-DNA duplex. And when we did this experiment over a spectrum of different pH environments, we saw that the wild-type Rho retained its activity throughout all the tested conditions, while the mutant Rho actually had a significant drop in activity that happened about pH 7.5 to 8. And when we look at another aspect of Rho activity, its ability to bind RNA, we saw a similar story, where at pH 7, there was really no discernible difference in the activity of the wild type and the mutants. But then at pH 9, there was a drastic reduction in the ability for the mutant Rho to bind RNA. So previous studies have illustrated that loss of Rho activity results in transcriptional read-through and an increase in antisense transcription. And so this indicates to us that, that likely this Rho mutation is going to lead to global changes in gene expression. And so next we wanted to do some pairwise competition assays to try to tease apart what conditions these mutations are favorable. And so we did pairwise competition assays after 1 day and 14 days in the unbuffered original evolved environment, media buffered to a pH 7, which is indicative of what the refeeding environment would be, and then finally buffered pH 9 condition, which the populations are spending the majority of the time, essentially 98 out of 100 days each cycle. And what we see is that with the Rho single mutant, there's a unique ability when nutrients are present after 1 day, but this ability is lost after 14 days. And you can see that this day 1 effect is most enhanced when the media is buffered to a pH 9. The YDCI single mutant strain was basically beneficial throughout all of the tested conditions. And then we also see that when we disrupt YDCI, it actually extends the Rho R109H advantage into that 14-day period, which is most indicative of the 14 days on the buffered pH 9 condition. And so given what we observe with the fitness and the biochemical assays, this indicates that there could be a change in intracellular pH, and really there would have to be a change in intracellular pH for the Rho R109H mutation to be in its mutant state. So you probably guessed what we did next. We tested intracellular pH. We did this using a single-cell microscopy-based method. We dyed cells with a dye called BCECF, and it has a altered or change in fluorescent intensity across a different range of pH. And when we looked at the intracellular pH of the wild type and single mutant strains, we really don't see any discernible differences between them with respect to intracellular pH. But what I hope you can appreciate at the bottom, the double mutant strain has a much more alkaline intracellular pH with respect to the environment. And when we graph the environmental pH across the different intracellular pH range, we see an elevation in the double mutant pH range, where it remains more alkaline with respect to the environment. And so this indicates that the double mutant creates this intracellular environment that would be conducive to see that phenotype of the alternate phenotype of the Rho mutants. So next we wanted to see how these mutations affected metabolism. And so to do this, we performed a metabolic array in a range of different carbon sources. This projector changed my color scheme, but it looks okay. And the heat map represents ratio to mean growth across each given resource. And overall, what we see is that the mutant strains perform better than the wild type. And also what we note is that the single mutant strains each provide access to a different subset of nutrients that are often retained in the double mutant strain. So I'm going to talk about 4 of those examples next. Those are alpha-ketogluteric acid, D-glucose 6-phosphate, L-aspartic acid, and pyruvic acid. And when we look at the transcriptome in midlog, we see that there is an increased expression in the transporters that import these nutrients. So that's very exciting. And what's also cool is these resources all funnel in directly into central metabolism or one step removed from central metabolism. And this is cool because previous studies that look solely at starvation have also indicated this broadening of metabolism. But the scary figure I have indicated with central metabolism in the middle, these are kind of far away from central metabolism. It takes a few steps to get to central metabolism. So here, in our case, the double mutant would be better suited when resources are replenished. And these overexpressed transporters in the double mutant are not just regular old transporters. They're actually ion-driven transporters. And so this appears that the increased intracellular pH of the double mutant confers an increased ability to transport nutrients. And so this brings us to a new hypothesis, because what's really interesting is that with the exception of aspartic acid, we really don't expect these resources to be available in large amounts in LB amino acid broth. And so we hypothesize that these double mutant cells are actually have an increased ability to feed off of what's being released from dying cells or necromass. And so I'm kind of taking that forward, and I'm measuring these resources in spent media in the cultures of our mutants over time. All I have to show you today is pyruvate, but what we see is really interesting, where at midlog in 36 hours, we really don't see a difference in the amount of pyruvate in the media, but after 5 days, we see this large increase in the amount of pyruvate in the double mutant cultures. And when we look at the population of our strains across the exponential phase, stationary, follow them through death phase, we see that the first rebound following death phase occurs, it's highly repeatable, and it occurs at the same time in our wild type and single mutant strains at day 6. But in the double mutants has that rebound a full day earlier. And so this suggests that potentially they're have that early rebound because they're better able to utilize those resources from the dead cells. And so I'm really excited about these data and looking forward to testing more of those resources. So that brings me to our model, where during the prolonged starvation period, we have an alkalized cell with respect to the environment that creates a positive delta pH across the membrane. And that, matched with the increased expression in the double mutant, is going to help drive in what little nutrients are available in the environment. This is going to be amplified when nutrients are returned, because the extracellular environment is going to go back to pH 7, but the double mutant is going to maintain an alkaline cell. And that's going to further increase the delta pH and further increase that drive to bring in nutrients right when they're available in the environment. Okay, so in summary, I showed you that delta YDCI mutations co-occur repeatedly with Rho R109H mutations in the 100-day feast famine environment, and that the Rho mutation confers loss of transcription termination activity at an alkaline pH, and then also the double mutant has a elevated intracellular pH range. All of this leads to an increase in the expression of particular transporters and better growth on intermediates of central metabolism that we would expect to be present in necromass. So these results indicate the nuance of studying starvation, because it appears that this refeeding aspect of our regime changed or constrained the evolutionary outcomes. And it also demonstrates the power of experimental evolution. Rho is it's an essential and highly conserved protein across many bacteria, and this experimental evolution was able to uncover a new function, and then also shed light on the function of previously unknown proteins. Oh, wow. And with that, I'd like to acknowledge our collaborators and the Behringer Lab, funding sources, and I think I might have time for maybe one question.
Thanks everyone for staying for the very last talk. Have a really good day. Thanks
to all the speakers. This has really been a fabulous session, I think.